Namaskar. Welcome to our channel by Gulab Sir for Wisdom Coaching Center. As a part of series of lecture, today's topic is Krebs Cycle. We already learned about first step of respiration, glycolysis. As glycolysis is a stepwise breakdown of glucose into pyruvic acid, it occurs aerobically as well as anaerobically. But further breakdown of pyruvic acid into carbon dioxide and water to release the full energy, complete energy, it occurs in mitochondria. Therefore, that Krebs cycle occurs in eukaryotic cells. So to learn Krebs cycle, that we have to have knowledge about glycolysis that we had learned in last week. Krebs cycle is a second step. Basically, it is a third step because there is an interconnection between glycolysis and Krebs cycle is carboxylative decarboxylation of pyruvic acid into acetyl CoA. And further that acetyl CoA undergoes cyclic breakdown to produce oxaloacetic acid is known as Krebs cycle. Let us understand what is Krebs cycle. Krebs cycle is discovered by Dr. Hans Kreb in 1937. Therefore, the name given Krebs cycle with his honor or honor to him. This cycle is also known as tricarboxylic acid cycle and citric acid cycle. To understand, so Krebs cycle is also known as TCA cycle and citric acid cycle. Pyruvic acid produced during glycolysis or as a part of last step of glycolysis undergo oxidative decarboxylation. It is so called decarboxylation because CO2 is released, 2 molecules of pyruvic acid that undergo oxidative decarboxylation to release 2 molecules of CO2. And since it undergo decarboxylation, oxidative decarboxylation, thereby hydrogen released from pyruvic acid combined with NAD to form NADH2. It occurs in presence of pyruvic acid dehydrogenase. That is what hydrogen is released. Therefore, the process is dehydrogenase. Pyruvic acid dehydrogenase singly do not catalyze this process, but there is a complex of pyruvic acid dehydrogenase complex. It is called as pyruvic acid dehydrogenase complex, which include magnesium ion, thiopyrophosphate, lipoic acid, and transacetylase Dear friends, this is very important to understand this pyruvic acid complex as a part of NEET exam. Question can be asked about. So here the first step, that oxidative decarboxylation of pyruvic acid, which produces acetyl CoA, coenzyme A. This is a product of intermediate state between glycolysis and Krebs cycle. This is a second step and Krebs cycle is a third step. But instead of saying this second step, we directly said that Krebs cycle is the second step. Now this NADH2 when oxidized during electron transport system in inner membrane of mitochondria lead to produce 6 ATP. So we have to consider right now. Later we shall learn about electron transport system to have transport of electron through inner membrane of mitochondria, while oxidation of NADH2 and FADH2 to release the proton and finally to produce the ATP molecule. But here this is what very clear to you, with the production of 2 molecules of NADH2, 6 ATPs are being produced. Now this acetyl CoA enter into the Krebs cycle. with the combination of oxaloacetic acid. Here this Krebs cycle begins with the condensation of acetyl CoA with oxaloacetic acid and addition of water. So here this condensation begins with the combination of oxaloacetic acid with acetyl CoA. In addition, that hydrogen, water is being added and CoA is being released. It is catalyzed by citrate synthetase. It is catalyzed by citrate synthetase. Remember these enzymes, that question can be asked in NEET, that one of the following enzyme catalyze the process of condensation process of acetyl CoA with oxaloacetic acid. Remember the process condensation. Krebs cycle begins with the condensation. So this is very important. Condensation. Even it mentioned in your textbook, very first sentence under the heading Krebs cycle, it begins with the condensation of acetyl CoA with oxaloacetic acid in presence of citrate synthetase with the addition of water and release of CoA lead to produce citric acid. See friends, every and each step is very easy. Once you practice this cycle twice or thrice, everything will be remembered, and what to explain, the things which you are saying, that is only explained. Every step you have to remember what is going on, and that is what step by step you have to remember. Now this citric acid undergo isomerization to produce the isocitric acid, but it has two steps. First step, release of water, catalyze in presence of iron. This also you have to remember, that it is catalyzed in presence of iron. So as a cofactor in NEET produces cis-aconitic acid. In presence of enzyme aconitase. Now this cis-aconitic acid again undergo hydration with the addition of water. Again it happen in presence of iron as a cofactor. Enzyme is same, aconitase, producing isocitric acid. Now this isocitric acid undergo decarboxylation, oxidative decarboxylation. But step by step, first it undergo dehydrogenation, that is 2 molecule of NAD undergo reduction to produce 2 molecule of NADH2, producing 6 ATP as a part of further breakdown of NADH2 into mitochondria, producing 6 ATP. These happen in presence of isocitric dehydrogenase. So here the enzyme name I mentioned, isocitric dehydrogenase, producing oxalosuccinic acid, Now oxalosuccinic acid undergo decarboxylation. Altogether, isocitric acid undergo oxidative decarboxylation, producing alpha ketoglutarate acid. So here oxidation, here decarboxylation. Together it is called as oxidative decarboxylation to produce alpha ketoglutarate acid. It is mentioned in your book. So please remember this one. This alpha ketoglutarate acid undergo oxidative decarboxylation. So how it happened? So to understand, oxidative decarboxylation is decarboxylation CO2, 2 molecule of NADH2 producing 6 ATP, 6 molecule of ATP. So alpha ketoglutarate acid undergo oxidative decarboxylation producing 6 molecule of AT and convert into succinyl CoA, succinyl coenzyme A. Here as this succinyl CoA undergo again oxidation by converting 2 molecule of GDP, 2 molecule of GTP. Here this is for succinyl CoA undergo oxidation producing this succinic acid. Succinic acid happen in presence of succinic thiokinase. Succinic thiokinase. Here this alpha ketoglutarate acid undergo oxidative decarboxylation. This process is oxidative decarboxylation again. This happen in presence of decarboxylase enzyme decarboxylase as well as dehydrogenase and alpha ketoglutarate acid dehydrogenase. See, as these two steps occur in presence of that iron as a cofactor, this decarboxylation occur in presence of manganese. This also occur in presence of manganese. So please remember iron and manganese. Now here this occur, this succinyl, this oxidative decarboxylation that occur in presence of manganese. Now that succinic acid which undergo oxidation in presence of succinic thiokinase, it occur in presence of magnesium. So see, these cofactors also has to be remembered as a part of NEET examinations. This question can be asked that which reaction is catalyzed by which enzyme in presence of which factor. Okay, now this succinic acid undergo oxidation where 2 molecule of FAD undergo hydrogenation to produce 2 molecule of FADH2 in presence of succinic acid dehydrogenase. Succinic acid dehydrogenase and form fumaric acid. Now fumaric acid undergo hydration, that is what addition of water to produce malic acids, and this malic acid undergo again oxidation producing 2 more NADH2 in presence of malic acid dehydrogenase. Malic acid dehydrogenase. So this is very clear to you that here also 2 more NADH2 undergo oxidation producing 6 molecule of ATP. Here each FAD when undergo oxidation produce 2 ATP, so total 4 ATPs are being produced. So see how many ATPs are total produced during this Krebs cycle. To understand, as this 2 GTP is equal to 2 ATP, so they are also considered in that total. So total number of ATP produced in Krebs cycle are 6 plus 6, 12 plus 6, 18 plus 2, 20 plus 4, 24 plus 6 ATP. So total number of ATP produced in Krebs cycle are 30 ATP. During aerobic respiration, that also because complete that process of glycolysis and Krebs cycle is a complete breakdown of glucose into water and carbon dioxide, and therefore take into consideration that number of ATP produced during glycolysis. So ATP produced during glycolysis, total ATP, total during glycolysis, that is aerobic glycolysis, aerobic, that is very important, are 8 ATP. So total number of ATP, therefore total number of ATP produced during complete breakdown, aerobic, complete breakdown of glucose during, that is aerobic respiration, are 38 ATP, but 2 ATPs are required to transfer NADH2 produced during glycolysis, that is when 2 molecules of phosphoglyceraldehyde undergo oxidation to produce 2 molecules of 1,3-DPGA, that NADH2 which produce in cytoplasm, they are supposed to transfer into mitochondria, consume 2 ATP. So NADH2 produced in cytoplasm during glycolysis in cytoplasm are transferred to mitochondria, consume 2 ATP, consume 2 ATP, hence net ATP produce during complete respiration are 36 ATP. Therefore, we used to write a chemical equation is 6CO2 C6H12O6 plus 6O2 plus 12 NADH2 plus 18 ADP. So here we understood net ATP produced during Krebs cycle and glycolysis, that is complete breakdown of glucose into carbon dioxide and water with the release of 36 ATP. So equation would be C6H12O6 plus 6O2 plus 10 NADH2. 2 NADH2 produced in glycolysis, 2 NADH2, 4 NADH2, here 6 NADH2, here 2 more NADH2 and here 2 more NADH2. So here total 10 NADH2s are produced and 2 FADH2, that is what, plus 2 GDP, that convert into 2 ATP later, lead to produce 6 CO2 plus 6 H2O plus 10 NAD plus 2 FAD plus 2 GTP, that is what included later in, plus now since this 2 GTP considered as a 2 ATP, so here we can write total 36 ATP, so 28 ATP, so that will be 36 ATP, so 34 ATP, so total 36 ATP. So here the equation is that what total equation is considered as that producing 36 ATP. So here we did understand Krebs cycle as a part of series of that lectures. We had learned glycolysis and now we learn about Krebs cycle. Point to be note that for NEET, that what is pyruvic acid dehydrogenase complex, that include magnesium, thiopyrophosphate, lipoic acid and transacetylase, that mineral factors which cofactor which catalyze the process are stepwise also to be remembered. Now here that this GDP produce GTP, this is substrate level phosphorylation to produce the ATP. So here you please remember substrate level phosphorylation as a separate part of phosphorylation, substrate level phosphorylation phosphorylation, that what to produce ATP. So this also can be the question of NEET. So here we understood Krebs cycle, as Krebs cycle has these three names: citric acid cycle, as it is the first product of Krebs cycle; tricarboxylic acid, as this is the 3-carbon compound; and Krebs cycle, as it was discovered by Sir Hans Kreb in 1937, and therefore with his honor the name given Krebs cycle. So dear friends, I hope if you did not understand repeatedly, you can see this video while explanation, you can repeat that event while explanation, so that you will get it clear what I am explaining. If it is not understood at a glance, then you can just repeat it to see that stepwise explanation. And even if you are in doubt, you can ask in comment section. I appeal you to like our channel and subscribe it. Also press bell icon so as to receive new videos as and when they are uploaded. Thank you.